Today you're going to be doing the Complete Personal Protein Translation Lab. So you're going to need to make sure to get one of these green copies from up at the front. It may be a different color for different years, but this year it's green. And you're going to want to have your past labs that we've done because you're going to need access to that full mRNA sequence. So here's where you need to make sure that you've been doing all of your labs correctly, because starting back here on our personal DNA lab, you had your entire unique sequence that you had written down here on this line. That coincides with your entire mRNA sequence. Okay, so this is actually the sequence down here, and it should be the full sequence, not just 12. So I'm not just looking for this tiny little sequence here. It should be your full mRNA sequence. If you have to, look back at your DNA lab. This is the sequence that you're going to have to write down here to get started. So be sure to start from number 1. Don't skip any of the steps. But I just want to show you how your lab is going to be set up today. So you're going to have some modeling pieces that you can use to set up to show the ribosome and the tRNA, and then your nucleotides that are on your tRNA and on your mRNA. You're just going to do a quick setup of that, just so that you can start to understand this process of translation and all the different pieces that are involved and what they do. So once you're finished with that model setup, which remember is going to come out of tins like this, you can just put all of that stuff away, and then the rest of the lab you're going to be focusing on building your protein out of Legos. So here's where you get to play with those Legos I promised you. You're going to use that genetic code that we've talked about to translate your mRNA codons into an amino acid sequence. Okay, so for instance, my first mRNA codon, as you can see, is AUG. Now this should be the same for everyone. So if I look up AUG in my universal code, I find the first letter here on the right, A, the second letter up at the top. I'm going to follow it to the box that's next to A and underneath U. And then I find G over here at the right and follow it back, and that codes for the amino acid methionine. Right? I'm going to do that for all of the codons in my mRNA sequence. So the next codon I look up is CAG. So I start here, C on the left, A on the top, follow it to the box that's next to the C and underneath A, and find G, and that stands for glutamine, GLN. So that's my next. So as you can see here, I've already used this genetic code to figure out my entire mRNA sequence. And now I'm going to build my protein. Okay, so this is going to be the last thing that I ask you to take a picture of here with all of your models together. So on Notability, you're going to show me your DNA model that we did in the first lab, the mRNA model that we did in our transcription lab, and now today you're going to have your Lego protein along with it. So I'm going to have to take my recorder here, and throughout the lab, we have different protein amino acids that are set up in each of their individual kits here. And you'll notice that each one of these has a label with it. So you'll see methionine, you'll see phenylalanine, proline, and as if we continue walking through the lab, you'll see the rest of ours, our amino acids, and I've tried to put them out in alphabetical order so that they'll be easier for you to find. So for instance, my first amino acid again is methionine. So the first amino acid I need is right here. So I'm going to take one of these, and this is the very first amino acid in my sequence. Now it needs to touch the next amino acid because remember, these are peptide bonded together. So now I need to go find my glutamine. And here we have our glutamine. So I'm going to take one of these, and it doesn't matter where I put it, I just need to decide where I want to have it. So I'm going to set him up here in the corner with his little leaves sort of fanning out. Okay, so that was my glutamine. Next I have phenylalanine. So I'm going to come back over here to where my phenylalanine is. Okay, here's phenylalanine. So I'm going to take one of these. And I do need to make sure that my phenylalanine is touching my glutamine. So I'm going to place him here. Okay, so remember that each amino acid gets peptide bonded to the next amino acid in sequence. Okay, let's see. Next, I need a threonine. Here is my threonine. So I'm going to grab one of those. I'm going to attach him. Now, you can get sort of clever with how you decide to attach these. So I could put it on top, I could put it in front, I could put it over here. So there's all sorts of possibilities, right? You are building your own 3D structure. So you do whatever you would like, right? Next, it looks like I need a proline. Here's my proline. And this guy's kind of big. I'm just gonna overlap this a little bit. All right. So again, you're making whatever shape you would most prefer. Okay. If that was my proline, the next in line, I have 2 serines. So here's serine. I get to have 2 of these because my proline, serine, serine. So let's say I'll attach one like so, and the next one we'll put here. All right, so as you can see, I'm starting to make this 3D structure of a protein, and when I'm all done, I'm going to take a picture and I'm going to add it to my Notability submission for this lab. When you're all finished, guys, please make sure that all of your proteins, your amino acids, get disassembled one at a time and they go back into their correct containers. If you can do this correctly, there may be a special prize for each of you.